So good morning. Actually, I'm not really sure if you guys are watching these lectures in the morning, but anyway, welcome to lecture number 16. So in our last lecture, we talked a little bit about how mutations, the forces that influence the frequency of mutations in the population, and that's actually a pretty good setup to today's lecture, where we'll talk about how you can use one type of mutation, namely neutral mutations, to ask evolutionary questions. So just a little bit about the figure. I'll talk about it more at the end. This is actually a figure that kind of tracks the migration of the COVID-19 virus basically throughout the world. And again, I'll say a little bit more about this. It's just a nice, pretty picture at this point. But I'll just talk very briefly at the very end of the lecture about how this figure was created and how tracking mutations can be useful. So, okay, in the last lecture, we talked a little bit about some of the principles of evolution, and I'm just going to very briefly review some of the concepts, mostly the concepts that set up today's lecture, because that'll help us to understand the first topic I'm going to talk about, which is basically the molecular clock concept. So from last lecture, I mentioned that mutations occur at every generation. Mutations over the generations accumulate. And I also mentioned that mutations fall into three broad categories depending on their effects on fitness. Neutral mutations is the ones I'm mostly going to talk about today. These are mutations that have no effect on the ability of a species to pass its genes on to the next generation, so they have no effect on reproductive fitness. The other two categories of mutations I talked about, which I'm not really going to be spending any time on today, are deleterious mutations and adaptive mutations. Again, since I just talked about this in the last lecture, I'm not going to spend too much time on it. I'm just mostly using this as a reminder to set up some of the things I'll talk about today. Of the three categories of mutation on the preceding slide, I also told you in the last lecture that most mutations fall into the neutral category. And the reason that's the case is because it turns out most of the genome, the human genome, is basically just codes for sequences that are not under selection. Only a small fraction of the genome actually codes for protein, and then a really tiny fraction codes for regulatory sequence, such as promoters and sites and introns that are involved in splicing, that sort of thing. Most of the genome, maybe about 95%, isn't really under selection. So you can have mutations occurring all throughout the genome, and it's really not going to have any effect on the fitness of the organism. Even mutations that map to the protein coding sequences won't really have any effect on fitness if they don't change the amino acid sequence. And that's also true to some extent with regulatory sequences where unless the mutation affects a specific site in the regulatory sequence that influences transcription or something like that, it's not going to have any effect on fitness either. Okay, so one of the conclusions I drew last time, I didn't really have time to go through this, but basically I said that because most mutations are neutral, we can actually use them to answer questions, really interesting biological questions. So, okay, let's go on to the next slide here. So amongst the types of questions I said you can use mutations to answer, one thing that we spent a lot of time in the first half of my part of the course talking about is how you can use mutations to do linkage studies. So for example, the SNPs that we used in linkage mapping or in genetic association mapping, um, these are just simply like for this example, as just one example of a type of neutral mutation. These are these polymorphisms that exist in the population. Most of these polymorphisms, these are just sites in the genome where you see differences between two different individuals in the population. Most of these polymorphisms don't really have any effect on fitness. They don't affect any of your traits at all. They're just places where in the genome where the sequences vary between different individuals in the population. And because they don't influence the traits, we can actually use them for experiments, like, for example, to map genes. So this is a case where we tried to map a gene in Drosophila, and there's just two different alleles at this particular site in the genome. And because most of these things are not under selection, there's no effect on fitness, we can actually use them to map genes. And the reason that's important is because if for example, one of these two alleles we were looking at to use to map a gene had some sort of fitness effect on the organism, such that, for example, maybe it influenced the likelihood of getting through embryonic development, then you wouldn't see them in equal proportions. And if you saw them at something other than equal proportions, that would influence your interpretation of a mapping experiment. So it's really important that they actually do not have any effects on the fitness of the organism. I also briefly mentioned that we could also use these neutral mutations to do evolutionary studies. I didn't really describe this in any detail, but that's what I'm going to talk about today. Basically, I showed this figure very quickly, and I just said you can also use inherited mutations to look at evolutionary relationships. But it's important that the types of mutations you use in these experiments are neutral. As it turns out, most mutations are neutral, so you can basically assume every mutation that accumulates over time, that you can kind of assume that they're all neutral mutations, even though some of them probably affect fitness. Okay, so we're going to start off by talking about this method to basically be able to start creating evolutionary relationships. And we'll use a very specific, we'll try and use mutations to answer a very specific question in this context. So, and again, the term that we're using here is the molecular clock. And so we're going to use mutations to say something about the evolutionary relationship of chimpanzees, which are the closest living relatives of human Homo sapiens. So if we look at the fossil record and we try and get an estimate of how closely related we are to chimpanzees, the fossil record basically says that humans and chimps shared an ancestor that existed maybe 5 to 7 million years ago. Okay, this is based on the fossil record. So we're going to actually try and independently estimate how long back this common ancestor of humans and chimps existed by just using mutations. Okay, and we'll talk about the concepts by which you do this. And this idea of, again, using mutations that accumulate over time to estimate evolutionary relationships and placing timelines on these evolutionary relationships, this concept is known as the molecular clock. We'll use mutations now to estimate the time since the last common ancestor of chimps and humans. So let's say we have this individual pictured here. We'll call this the most recent common ancestor of humans and chimps. And let's imagine at the very first point where humans and chimps started to diverge from our last common ancestor. Let's say this is the very first generation, right after the two different species started to diverge from one another. What's going to happen here is after even just one generation, the offspring are going to actually acquire a new set of mutations, because basically every single time the genome is replicated, a small number of errors occurs. Now, the machinery for replicating the genome either during mitosis or meiosis, the machinery is very, very precise. It rarely makes mistakes, but the human genome is really huge. It's 3 billion base pairs in length. So even if you only make a mistake once every, say, million base pairs, that's still going to be a lot of mistakes, right? And actually the machinery that basically replicates the genome makes mistakes at a lower frequency than that. So at any rate, even after one generation, you're going to have acquired a set number of mutations, just because the process of copying the genome is somewhat error prone. So after the genome is replicated, after one generation, each branch in this lineage is going to acquire a new set of mutations. So one set of mutations is going to be acquired in one of the two lineages, and another set of mutations is going to be acquired in this other lineage. But then if we so that's 2x new mutations in total. If you compare the genomes of these two organisms, that'll be 2x new mutations that will differentiate the genomes of these two individuals right after the split from the last common ancestor of humans and chimps. And then if we go many, many generations and we finally end up with chimps and humans, after n generations, the number of generations that have gotten us to the present time from our last common ancestor, that's what n represents, we're going to have acquired n times 2x new mutations. That's how many differences we're going to see between humans and chimps if we compare our genomes. So again, a simpler way of putting this is that after n generations, there will be n times 2x differences between the chimp and human genome sequences. And again, it's simply because at each generation, new mutations are passed on between the parents and the offspring, okay? And if we just assume that x is the number of mutations that are acquired in each lineage at each generation, then it's going to be the number of generations times 2x, because there's mutations being acquired through both lineages. So that's why we have the 2x differences between the chimp and humans genome sequences. So I've now given you enough information that we can actually sort of use this to calculate when the last common ancestor of humans and chimps existed. But there's a number of things we need to know to really do this calculation. So again, the question is, what do we need to know to answer this question? And basically, there's two fundamental things we have to figure out. One thing is we need to figure out what x is. basically, and again, X is the number of new mutations that are passed on by the parents to their offspring at each generation. And the other thing we need to find out is, how many differences there are now between humans and chimps, because the number of differences between the two of us, those are all the mutations that we've acquired since the time we split off from our last common ancestor. All right, so what I'm going to do now is I'm just going to put this more in mathematical terms. Again, after n generations, there's going to be n times 2x differences between the chimp and human genome sequences. So just putting this in a more mathematical context, I'm going to say the number of differences in the genome sequence between humans and chimps is going to be equal to the number of generations since our last common ancestor times 2x, where x again represents the number of mutations that are acquired from our parents at each generation. So I'm now just going to use algebraic manipulation to rearrange this equation. I'm just going to divide both sides of the equation by 2x. And that gives me n equals the number of differences divided by 2x. So we can get the number of generations just by taking the number of differences in the human and chimp sequence and dividing that by 2 times the number of mutations that are acquired at each generation from either of the two lineages. So the number of differences is easy to obtain because we have the human genome sequence. It's already been obtained. We've also obtained the chimp DNA sequence. And so all we have to do is create an alignment between the two of them and just look for differences. And basically when we do that, we look for differences between the two. We tally that up over the entire genome. What we find is, in fact, the human-chimp DNA sequence similarity is 98.8%. So the size of the chimp and human genomes is basically the same. They're both about 3 billion base pairs in length. So if we want to know the total number of differences between human and chimp, we just take the difference in similarity, which is 1.2%, multiplied by the length of the genome, and that tells us that there are 35 million differences between the human and chimp sequence. So that means over the time since our last common ancestor, in both branches together, we've acquired 35,000 mutations that differentiate the two genomes. Okay, so the question is, how do we obtain X, the number of mutations that are acquired from our parents at each generation? So the way we do that is we have to sort of basically compare the sequences of the genome from a set of parents and the offspring, and then just tally up the number of differences that were acquired by the child that are not present in either parent, okay? So just reading what I've said here, to calculate the number of mutations that are passed down each generation, we need to compare the DNA sequence of a mom, dad, and child and look for positions in the child that don't match either parent. This is also known as trio sequencing. People do this a lot to actually also identify like if a child has a disease, neither parent seems to have the disease, doesn't look like it's necessarily some recessive disorder that had been passed on through the families. Oftentimes that means that there's a new mutation that was acquired by the child, and the mutation occurred in one of the two parents' gametes. So one way to identify that mutation is just by comparing the sequences of the mom and the dad and the child to identify any new mutations that are present in the child. And then that gives you a set of candidate genes that might represent the gene that's responsible for the disease in the child. Okay, so people have done this sort of thing. And again, you're just asking how many differences the child have that are not present in either parent. It turns out the answers, we can get the answer to this, but it really actually depends on paternal age. So it's not just a uniform number independent of age. It actually really does depend on the paternal age. This is a picture of Evan Eichler. He's a faculty member in the Department of Genome Sciences here, and he's actually one of the ones who's actually done some of this work to kind of estimate how many new mutations are passed on from parents to their offspring. And what's plotted here on the right is a graph, and there's if you look at the left side of the graph, it's showing us the number of mutations that are passed on to the offspring from both mom and dad anywhere in the genome. On the right is the number of mutations that are present in the exome, which is just a small fraction of the genome, right? Just the expressed portions. So that number is going to be a lot smaller. And on the bottom, along the x-axis, is paternal age. And you can see basically what happens is paternal age increases, the number of mutations that are sort of passed on from parents to offspring increases over time. Okay? Turns out that this is a specific feature of paternal age because Females transmit about 25 new mutations to their offspring regardless of their age. So this is a male-specific phenomenon. So it doesn't matter whether the mother is young or old, she'll pass on about the same number of mutations. But with males, they pass on more as they age. I'll talk a little bit about why that is in just a second here. I just want to mention an aside. It's it's a sort of an interesting aside, I think, is that it's become increasingly clear that paternal age correlates with the frequency of certain diseases in their offspring. And I'm just going to show you one example. There's lots of these, though. This is an example that sort of talks about the relationship between paternal age and schizophrenia risk. And what it basically shows you is that depending on paternal age, as the father gets older and older, the likelihood that that father's offspring are going to develop schizophrenia increases. And the reason this is the case is it's thought that it's because as the father gets older, he's passing on more and more mutations to their offspring. And it turns out schizophrenia is definitely a genetic disease. There might be an environmental component to it as well, but it's clearly, it's also partially caused by genetics at the very least. And so the more mutations you pass on to the offspring, the more likely that your offspring are going to inherit a disease that's associated with the genetic component. And so schizophrenia is one of many diseases that falls into this category. There's a bunch of others as well. So for example, autism. Also, the frequency of having a child with autism correlates with paternal age. It's like twice as high. The frequency is twice as high with fathers that are like over the age of 40, for example, of having a child with autism. Bipolar disorder, and there's a number of neurodevelopmental disorders that show this trend as well. So again, all of this is thought to be because as the fathers get older, they pass on more and more mutations to their offspring. Whereas with maternal age, these trends are typically not seen because mothers pass on roughly the same number of mutations regardless of age. So why is this the case? The reason this happens is because of differences between males and females and how their germline basically develops. And basically what's shown here is that the key difference here is, I'll just summarize it in one sentence, is that there's way more mitoses in males, and the number of mitoses that give rise to the germ cells are increased by age dramatically, but there's way more in males than there are in females. So if you see the story in females on the left, there's roughly 22 divisions that give rise to primordial germ cell during fetal development. And then basically on roughly a monthly basis, one of these germ cells is selected and goes through meiosis all through life, basically. So essentially there's only 22 mitoses required for germ cell formation in females. But in males, the story is really quite different. Males undergo about 30 mitoses during fetal development to form the germline, and then another 190 mitoses occur in young males. But then by the time males become about 40 years of age, the germ cell population has undergone about 650 mitoses. And the reason for that is males make many, many, many sperm cells, and they're making them all through their life. And so the germ cells, the germ stem cells themselves have to undergo many, many replications to produce all these sperm cells. So basically, if you actually look at the difference here, it's actually surprising that the number of mutations passed on from older fathers is only maybe about 2 times more than females. You might think, looking at this, that the number of mutations passed on by males would be more in the order of 20 times more. Because of the number of increased mitoses. And again, the reason for the increased number of mutations that are passed on is because, again, it comes down to the same fundamental thing. The reason we pass mutations on in the first place is because copying the DNA, even though the enzymes that do that are very, very, very good and rarely make mistakes, they still make mistakes on occasion. And if you have to go through a number of mitoses to get to the point of making a germ cell, the more mitoses you have to go through, the greater the odds you're going to basically have a mutation occur. Okay, so getting back to the question here. So as I mentioned, paternal age correlates with the offspring mutation frequency, but let's just think about this in evolutionary terms. Now, for sure, in more recent years, the trend has been for fathers to be a little bit on the older side. But over evolutionary times, probably the age of fathers was more about the age of 20, so we're going to use that for our calculations. So over like historic times, fathers were probably on average about 20 years of age. And if you look at the 20-year age point, that means about 50 new mutations are passed on by the parents. So about half of those get passed on by females, about 25 of them, and about half of them get passed on by the father, another 25. So we're going to use 50 mutations in our calculations to kind of calculate the divergence time from the last common ancestor between chimps and humans. All right, so 50 new mutations are passed on each generation by mother and father to their child. So now we have everything we need to do to finally do this calculation we were after, trying to figure out the time since the last common ancestor of humans and chimps. So again, how many new mutations per generation? It's 100. Now I know I just said on the previous slide, it's 50 new mutations per generation in humans, but there's another 50 new mutations that occur in the chimp lineage as well. So basically the way of putting this is that at every generation, the number of differences between humans and chimps is going to be 100, because 50 new mutations is going to occur in the human lineage, and 50 new mutations is going to occur in the chimp lineage. So there's going to be 100 new differences between humans and chimps at each generation. Okay, so now we take that value and we can use that to calculate the number of generations since the last common ancestor of humans and chimps. We do that by taking the total number of differences between the human and chimp DNA sequence, which is 35 million, divided by 100 new mutations per generation, and that gives us 350,000 generations. And then the last thing we need to know is what the generation time is in humans and chimps. And people argue about this a little bit back and forth, but most people agree that the generation time in both humans and chimps is roughly about 20 years. So now knowing this, we can use this information to calculate the time since the last common ancestor. It's been 350,000 generations since the last common ancestor, about 20 years per generation. So that gives us a value of 7 million years. So using mutations, we calculated that the last common ancestor of humans and chimps existed about 7 million years ago. And as I said when we began this analysis, that the fossil record suggests that the last common ancestor existed about 5 to 7 million years as well. So using this mutational approach, or the molecular clock approach as it's called, actually works pretty well. So not too bad. All right, so now on the next slide, I'm going to sort of test your knowledge, your understanding of what I just talked about by asking you to do a calculation. So the question we'll ask here is, how long since the last common ancestor of the African forest and savanna elephants? So I know probably typically people talk about African elephants as almost like they're one species, but it turns out they're actually there's two different African elephant species, savanna and forest elephants. And this just summarizes some of the differences between them. Probably the most obvious differences is that the savanna elephants are much, much bigger than the forest elephants, but they have a number of other differences that are just briefly summarized on this slide. They also have a very different geographical distribution, which is typically one of the things that leads to speciation, is when you have one species and one member of that species gets isolated from another member of the species, and over time they develop different characteristics. This slide shows you both the past distribution of the two different African elephants and the present distribution. It's a little bit of a sad figure. The forest elephant tends to occupy more equatorial Africa, whereas the savanna elephants tend to occupy more the northern and southern Africa more. Again, the range has been greatly reduced as a result of mostly human intervention, particularly for the savanna elephant. Okay. So, all right, getting back to the question here, how long ago did the last common ancestor of the savanna and forest elephants exist? And I'm going to give you a little information to help you answer this question. The genome size of both the savanna and the forest elephants is about 3.7 billion base pairs. The average generation time for both species, we're just going to call it 25 years. The number of mutations transmitted per generation in each species is 60. So 60 in the African forest elephant and 60 in the African savanna elephant. And then if you compare the genome sequences of savanna and forest elephants, they're about 99.5% identical. Okay, so using this information, I'm going to let you calculate the time since the last common ancestor of the savanna and forest elephants, and I'll let you choose between any one of these four answers here. So again, if you want to think about this on your own, this is the time to basically stop the video, go ahead and try and figure this out. and then I'm going to walk you through the numbers after you restart the video. Okay, hopefully you got the answer as about 4 million years. And just to quickly walk you through the numbers on this, basically the number of differences is equal to just 0.5% different, right? So I'm just going to do these calculations. The number of differences in these two different elephant species is 0.005 times the size of the genome. So it's about 18.5 million differences between the two of them. The number of generations is equal to the number of differences divided by the total number of mutations acquired at each generation. And again, I'm just taking the number of mutations transmitted per generation in each species. I have to double that because there's going to be 120 new mutations at each generation, because 60 come from one lineage and 60 come from the other lineage. And that gives us 155,000 generations since the last common ancestor. And if we're going to take the average generation time as 25 years, we just multiply 25 times the number of generations, and that gives us the time since the last common ancestor. And that should come out to roughly 4 million years. I know it's a little bit less than that, but it's roughly 4 million years. All right, let's go back to a previous slide for a moment, because I used this and I made an assumption you understood what this is. This is basically a relationship tree. It has other names as well. It can be called an evolutionary or a phylogenetic tree as well. I think probably most people are familiar with these. You've seen these before. I think I may have actually even presented some of these in the class before today as well. So it turns out Charles Darwin was the first to come up with this idea. This is actually a drawing from his notebook where he basically was drawing the tree that tried to describe how the different finch species could have derived from one another, how they might have had a common ancestor. So he was the first one to come up with this basic idea. And the tree on the left, the one that's based on the fossil record, the tree that Darwin was trying to draw, these are basically based on the one on the left is based on the fossil record. The one that Darwin was trying to draw was based on morphological criteria. And in fact, traditionally, most evolutionary trees do rely on morphological criteria. So I'll describe the basic approach that people use for trying to create genetic trees that are largely derived from morphological criteria on the next slide. So a typical way of creating a phylogenetic tree is by, for example, comparing observable traits between the organisms you're trying to group together. So I'll just give you a really simple example of this. Let's say we're considering the species that are showing here: deer, lamprey, salmon, eagle, and alligator. I wonder, maybe alligator has two L's. I might have misspelled that. Don't worry about that. Anyway, let's just say we're going to compare them in terms of the traits I've got listed here, where they've got feathers, fur, lungs, gizzard, and jaws. And so we'll just look at deer and we'll just check off the things that deer has. So deer have fur, they have lungs, and they have jaws, right? And then we're going to take the same approach for all the different organisms that are shown here on this slide, and we're just going to put a check mark if they have any of these features. And then the way people typically group them together in an evolutionary sense is by looking at how many characteristics they share in common or how many differences they have. And so if we look at this, we'll start with the latter. We'll basically say the group that's probably most distantly related to all the others on this group are the ones that share the least number of characteristics. And so the one that jumps out at us is lamprey. So it's probably most distantly related to the other species simply because it doesn't have any of the characteristics that are shown in this table. And then we could sort of go from there to say what's the next most distantly related to the others. That would probably be salmon because it only has one of the characteristics. And you just keep going this way. And then based on this information, you can draw basically you can draw a phylogenetic tree based on just comparing these different traits, basically. Okay? And the way that we create these genetic trees using DNA sequences is essentially the same. But we're using the DNA sequences and comparing DNA sequences and looking at how similar or different they are to one another. And I'm going to walk you through in the next set of slides one approach for creating a relationship tree using DNA sequences, mostly because I just want to make sure that the logic is clear in how these trees are constructed. But as I'll mention at the end of that whole procedure, there are many different ways that people use to make these genetic trees. And the one thing that I do want to make sure that I'm clear about is I want you to understand the logic by which these genetic trees are actually created, but I'm not going to give you anything on an exam that's as difficult as the example I'm going to show you on the next few slides, even though the example I'm giving you on the next few slides is actually much simpler than the kinds of things most people try and sequences people try and create genetic trees with under normal circumstances, okay? All right, so again, I'll try and make these points clear on the next slide to reassure you, okay? Okay, so I'm giving you a simple example of how to construct an evolutionary tree using mutations. So let's imagine we have sequences of the same gene from 6 different species, or what's more commonly done is people are comparing entire genomes or many genes together at the same time. But again, I'm just starting off with a very simple example, although, as I mentioned, this is a very simple example. It's actually way more complicated than anything I will give you on an exam, because it would take you a long time to do what I'm going to walk you through on this next set of slides, all right? But I'm just mostly trying to convey the logic that's used in creating these phylogenetic trees using mutations or genetic information, okay? All right, so this first step you need to do is, let's say we've got these 6 sequences that derive from 6 different species. We're just labeling 1 through 6. So step 1 is you need to create alignment between them. So let's do that. Here's the alignment of these 6 sequences, 1 through 6. And then what we're going to do is we're going to start comparing the sequences in a pairwise fashion. And ultimately, I'm going to have a table in here that's going to help us keep track of all of this. So for example, let's compare the sequences of number 1 and number 2. And we'll ask ourselves, how many differences are there between them? Okay? So how many differences between sequence 1 and 2? And now I'll highlight those differences. Okay, so if we count these all up, I think that there's 9 differences between sequence 1 and 2. So the differences, again, are highlighted, not the similarities. Now I'm going to have a table in my place up here on the right, and we'll try and keep track of all the steps I'm going through here. Okay, so we're going to keep track of all the pairwise differences. As I said, if we compare sequence 1 and 2, I think there's a total of 9 differences. We're just going to enter that value in the table here. And then we're going to move on. We're going to ask how many differences are there between sequence, say, 1 and 3. We'll do that one next. So I'll highlight the differences there. In this case, there's 2 differences between sequence 1 and 3. So I'm just going to enter that value in the table. 2 differences. And then we just keep going in this fashion until we have this whole table filled up. So I'm going to leave it to you if you want to do that to make sure you understand what I'm doing here, but I'm not going to walk you through all the steps here. I'm just going to fill out the rest of the table here. All right, so the table's all filled up. Now we're going to move on to the next step here. So step 3 is to identify the sequences with the fewest number of differences. So we're just kind of looking at the table here, and we're sort of saying which sequences in this table have the fewest number of differences. And if you look at this, if we compare sequence 1 with sequence 3, you'll see that there's only 2 differences there. But also, if we compare sequence 2 and 5, you'll see there's 2 differences there. Okay, so let's highlight those sequences. So again, sequences 1 and 3, they only have 2 differences. Sequences 2 and 5 also only have 2 differences. All right, so all the other ones in this table, the number of differences is greater than that. All right. And then the next step now is we're going to create the first groupings on our genetic tree. All right, so the first grouping is going to be 1 and 3 because they only have 2 differences between them. So we're going to put them together. They're most closely related. That's what this part of the tree is going to represent. But also sequences 2 and 5, they're also roughly equally related as 1 and 3. So that's going to be our other grouping. All right, 2 and 5. So now we have our first 2 groupings on the tree. Now the next step is going to be comparing the groupings in a pairwise fashion. So this idea might be a little bit tricky to follow, but just listen carefully. It's really not that complicated. I'm going to put another table in this figure so we can kind of keep track of the situation. But what I'm doing here now is we're going to start with group 1 and 3 first. So I'm putting 1 and 3 together in the table, and we're going to sort of come up with the average number of differences between 1 and 3 and the other sequences that are in this table. So how many differences on average between group 1 and 3 and 2? We'll start with that, all right? We're going to go to this upper table to make that calculation, okay? So what we're going to do is compare 1 with 2, and we're going to compare 3 with 2, and we're going to average those 2 values, all right? And those are highlighted here. So this is a comparison of 1 with 2 and 3 with 2. And on average, what we see is there's 9 differences together. So we're just going to put that average value in the table on the bottom, okay? And the average value happens to be 9 because it's the number of differences between 1 and 2, and 2 and 3 is exactly the same. They're both 9. And then we're going to go on and we're going to compare between group 1 and 3 and 4. And we're just basically using the same logic. We're going to look at how many differences there are between 1 and 4 and between 3 and 4. And I'll highlight those in the table above again. So that's the number of differences between 1 and 4. There's 4 differences. And between 3 and 4, there's 5 differences. So the average of those two is now 4.5. And we'll put that in the lower table. Okay? And then we just keep doing the same thing, and I'm just going to fill out the rest of the table with all this information. Some of the stuff that I put down here I just borrowed from the table above, okay? Like all these values here, because these are not grouped in any way. So I'm just taking these and moving them from the upper table to the lower table, all right? And then in the next slide, what I'm going to do is I'm going to take this lower table, I'm going to move it up to the top, and then we're going to create a new table down below, and we're going to kind of go through the same process, but we're going to do it with the group 2, 2, and 5. Okay, so I just moved that table up, and now we're going to go through the same logic with the grouping 2, 5. All right, so here's our new table, and now we're going to calculate how many differences on average there are between 2 and 5 and 1 and 3. And the way we do that, I'm going to highlight the ones that we're going to be comparing here. It means we're going to look at the number of differences between 1 and 2 and 3 and 5. And those are highlighted here. And you can see in both of those comparisons, the values are 9. So the average number of differences is 9. And I'm just going to take that value and put that in the table below. All right. And then I'm just moving these other ones down to this table below because what I'm doing here is just borrowing that information and just moving it down to the lower table. We don't have to redo those calculations because we've already done them. Then we're going to look at the average number of differences between group 2 and 5 and 4. All right. So I'll highlight those values in the table above again. So I'm just all I'm doing here is I'm comparing the sequence 2 with 4, and I'm comparing sequence 5 with 4. Okay. And the average number of differences between those is again 6 because it's basically 6 for each of them. And I'm putting that value down here. And then I'm just going to fill in the rest of the values here in this table. Okay, but I'll let you guys make sure you follow along with what I'm doing. And then at this point, what I do is I'm going to now create a new grouping in our phylogenetic tree. I'm going to add the sequence of the fewest changes to the tree. So if you look at this down below, what you can see is the grouping with the fewest, the sequence of the fewest changes that I can add to the tree is 1 and 3 and 4. There's only 4.5 differences. So that's telling me that sequence 4 is more closely related to 1 and 3 than it is to anything else. So that's the new grouping is going to be connected to the sequences 1 and 3, like so. All right. Okay, so now I'm just going to take this table on the bottom. I'm going to move it up and replace the one at the top, and then we're going to add another table, and we're going to finish off the tree. All right, so now we're going to finish this off. So the next thing we do is we're going to determine how many differences on average there are between 1, 3, and 4, and 2 and 5. I'll highlight the comparisons we need to make in the table above. So the highlighted comparisons are what I'm showing you here. So I'm looking at 1 and 3 and 2 and 5, and I'm looking at 2 and 5 and 4. Okay. So the average number of differences between those is going to be 7.5. So 9 plus 6 divided by 2 is 7.5. And then I can take the other values in this table and just move them down. Oh, I guess I have to do this calculation too. Between 1, 3, and 4, and 6. So I just have to average those two, and the average of those two is 10. And the last value I'm just moving down to this table, okay? All right, so now we have all the information we need to have to actually put on the last few groupings in the table. So if we look at which sequences have the fewest differences, it's grouping 1, 3, and 4, and 2 and 5. So now we can connect these two groups together, because these are the most closely related to one another. So we connect these together, and then the ones that sequence 6 has 10 differences with both of these groups, so it's going to be the most distantly related to all these, all right? And so this is going to be an outgroup that's connected at the very top to all of these other groups. Okay, because 6 has the most differences with these groups, so it's the most distantly related to all these other sequences. And that gives us our final phylogenetic tree. And basically, again, the phylogenetic tree is just based on comparing the number of differences in sequences between all these different sequences we're comparing. All right, just a few things I want to summarize here. As I mentioned before I started all this stuff, there are many different methods for creating evolutionary trees. The method that we use in this case is called the unweighted pair group with arithmetic mean method. But again, there's many, many methods that people use to create evolutionary trees. Turns out that the logic for all these different methods is basically the same. And it comes down to this: the more similar a pair of DNA sequences are to one another, the more recently they shared a common ancestor. That's the basic idea. Now let's just talk a little bit about how to interpret an evolutionary tree. I think you already know this, but I'm just going to make sure the points are clear. So, okay, basically, if we look at this, the evolutionary tree is supposed to represent time, where the past is represented on the left with respect to this tree that's shown here. The present is reflected on the right. And each of these points, these nodes, these are considered ancestors. Like each of these points that I'm showing you here are the last common ancestor within the groupings. I'll just make that more clear in just a minute. All these different sequences basically at the tips of the tree are the descendants. And so if we looked at these nodes of the tree, for example, position A, that would be the most recent common ancestor of species 1 through 6. Point E in the tree is the most recent common ancestor of species 2 through 5. Okay, so these ancestors are no longer with us anymore, right? These are like evolutionary relics from the past. But I'm just trying to make these points because I'll be using these when I talk about evolutionary trees. Okay, so now just to make sure you understand how to create these evolutionary trees, I'm going to give you a much simpler case and see if you can't distinguish which is the correct evolutionary tree based on sequence comparisons between the organisms we'll be talking about on the next slide. Okay, this is the question here is, what is the evolutionary relationship between humans, chimps, bonobos, and gorillas? And what I'm going to do is I'm going to fill out this table with pairwise comparisons of the genome sequence similarity between these different species. And we've already talked about one of them. I already talked about the human-chimp genome sequence similarity, and I told you it was that human and chimp genomes are 98.8% identical. And I'll just fill out the rest of these comparisons because it turns out the bonobo sequence has been obtained, the gorilla sequence has been obtained, so we can make these comparisons between these other species as well. And when we do that, we get the values that I'm showing you in here, okay? Now, sort of based on this information, I'm going to ask you which of the following trees appears to reflect these sequence similarities between these organisms. So I'm going to give you 3 choices here to choose between. And you can look at these sequences and try and figure out which of these trees is consistent with the percent difference in each of these sequences. What I'm showing you here is the percent identities, not differences. The differences would just be by taking 100 and subtracting these values from it, all right? So I want you to look at these numbers and see if you can figure out which of these trees makes sense given these numbers. All right, you can stop the video here and I'll just give you the answer, and then I can even help you to understand the logic behind this. All right, hopefully you picked sequence C as the correct answer. And what you can do is if you look at this and follow the same logic I used in the previous slide, if you look at this and say which of these sequences is the most similar, what you'd see is that it's the bonobo and chimp sequences that are most similar to one another. So that tells us right away that bonobos and chimps are the first grouping in our tree. They're the ones that have the most recent common ancestor of all the species that are present on here. So they're 99.6% identical. And there's 2 trees that are consistent with this finding. This tree number B, because chimps and bonobos must have the most recent common ancestor of all these species because they're most similar to one another. And this tree number C is also consistent with that. However, tree A is not consistent because, as you can see, if you compare the bonobo sequence to humans or the chimp sequence to humans, it's not as similar as the bonobo and chimp sequence comparisons are. So this tree does not make any sense. All right, so now let's look at the table a little bit more to figure out which of the two tables that are remaining, B or C, is the correct one. So the next most closely similar sequences are, if you look at the human and chimp and human and bonobo sequences, those are the two most closely related to humans. The gorilla sequences are more divergent from both humans, chimps, and bonobos. So the next grouping in the tree would have to be humans. So that would be tree C. Tree B is not consistent with, because gorillas are clearly more distant related to chimps and bonobos than humans are. So tree B would not make any sense based on that finding. So anyway, so does it seem odd to you that we humans are actually more closely related to chimps and bonobos than gorillas are? Would you expected humans to be more divergent? If that was your intuitive sense, you'd be wrong about that, because it turns out we're more similar to chimps and bonobos than gorillas are. It may not seem like that would be the case, but that's how it is. All right, so C is the correct answer. All right, now let's use some of the principles we've been talking about to talk about what the origin of anatomically modern humans are, okay? And there's been two theories that were out there for a very long time to explain the origin of anatomically modern humans. When I say anatomically modern humans, I'm talking about Homo sapiens, okay? So there's the two models are the multi-regional model versus the out of Africa models. So what's shown on the left is the multi-regional model. And basically what this said is that humans arose, modern humans or Homo sapiens arose independently in different parts of the world, completely independently from one another. So over a long period of time, modern humans arose completely independently from one another in different regions of the world. All right? And the out of Africa model said, reaches a different conclusion. It says that modern humans arose first in Africa, and then they spread out from Africa to the rest of the world. Basically, they migrated out of Africa and spread throughout the world. So which of these two models is correct? Since we're talking about mutations, we're obviously going to use mutations to distinguish these two models. And the initial approach that was used to distinguish these models was by collecting samples from individuals across the globe and sequencing portions of the mitochondrial DNA. And then what they did is they compared sequences in group by similarity, reconstructing as simply as possible. And the term that's used to do this is parsimony, the simplest possible explanation, essentially, the genetic tree that could account for the results. All right. Now, I can't sort of walk you through all the steps involved in creating this genetic tree, because the sequences that they were comparing were much larger. There were many more differences than the simple comparison we just made in my simple example. So it'd be way too complicated and would take way too long. So I'm just going to show you talk about the logic a little bit, and I'll show you the results of that analysis. So first question you might be wondering is, why use mitochondrial DNA? All right, let's talk about that for a minute. So I think this is a pedigree that looks similar to something you've probably seen in the first half of the class. This shows a mitochondrial inheritance pattern. And basically the pattern that you see here is that the mother's children all inherit the trait that you're looking at, and the father's children never inherit the trait. And the reason that's the case is because mitochondrial DNA is inherited strictly from mom. So again, in addition to the nuclear genome, we have one other genome in humans, which is the mitochondrial genome. And the reason that mitochondrial DNA is just strictly inherited from the mom is because the mom packs lots and lots of mitochondria into the egg, but the sperm does not bring any mitochondria with it. It just brings the nuclear genome. So all the mitochondria that you inherit, all the mitochondrial DNA, it always comes from mom and strictly from mom, never any from dad. So the fact that mitochondrial DNA is strictly inherited from mom makes it easy to track the maternal lineages. You can track the mitochondrial DNA transmission. Another feature that makes mitochondrial DNA very useful is mitochondrial DNA does not recombine. Okay, and as it turns out, these features of the mitochondrial genome are also evident in the Y chromosome, because the Y chromosome also doesn't recombine, other than that very tip of the Y chromosome that will pair with it's the pseudoautosomal region of the X chromosome. Most of the Y chromosome does not recombine, okay, except for a tiny sort of little bit of it that happens during meiosis, just to kind of hold the chromosomes pair during meiosis. But the fact that these chromosomes don't recombine makes them very easy to track over many generations. And Unlike the nuclear genome, which I talked about in a previous lecture, I sort of told you that it's not unusual to sort of, if you go back, say, 10 generations, to not even see any DNA in common between an ancestor that's 10 generations back, because the nuclear genome recombines so frequently, and based on the recombination the frequency of recombination and the random pattern of segregation, you're just going to have very little in common with somebody who's even maybe, like I say, 10 generations back. But basically, you're inheriting exactly the same mitochondrial genome from somebody 10 generations back if you track that mitochondrial genome just through maternal lineages, right? It's not going to come from the males, but you can track it all the way back 10 generations, and you're getting exactly the same mitochondrial genome. It's going to have acquired a few a few mutations over that period of time, but by and large, it's exactly the same mitochondrial DNA molecule, having undergone no recombination events. And what I'm saying about the mitochondrial genome is exactly the same for the Y chromosome. So these make it very easy to sort of create genetic trees and to track lineages. So when people took this mitochondrial DNA and they basically tried to create a genetic tree, the results of that looked like what I'm going to show you on the next slide. All right, so this is the evolutionary tree that was created using mitochondrial DNA sequencing. And they sequenced portions of the mitochondrial genome from populations all over the world. And since this is the lettering here is very small, I'm just going to highlight the key thing here. Is you can see that the older branches of the tree are all African in origin, and the younger branches of the tree are all non-African. And basically, the fact that that's the case indicates that all these mitochondrial DNA molecules derive from populations that were present in Africa initially. So this was supported the idea for the out of Africa model of human evolution, which basically established that modern humans, began in Africa, and then they left Africa after they at a much later point. So, and then based on the mutation frequency on the mitochondrial genome, this is essentially just using the molecular clock model, and I'm not going to go through this in any detail. They also estimated that the African origin goes back about 150 to 200,000 years ago. So basically what that tells us is that everybody on the planet shares a common maternal ancestor that they can trace their mitochondrial DNA back to, who existed in Africa maybe 150 to 200,000 years ago, okay? So that's true for everybody on the planet. You can trace your mitochondrial DNA back to a common maternal ancestor that existed maybe 150 to 200,000 years ago. And then subsequently, people did the same kind of analysis with the Y chromosome, because again, the Y chromosome has the same characteristics that allows us to be useful in this kind of analysis. And they got exactly the same result, where they showed that the oldest lineages were all African lineages. And again, it supported the idea that that there was an African origin of all modern humans, and the timeline of the African origin was about the same. It says that all males on the planet can sort of trace their Y chromosome back to a male that was present in Africa maybe 150,000, 140,000 years ago, something like that. And you'll actually see terms in the literature, even the scientific literature, that reflect these situations. You'll hear this term mitochondrial Eve, which is supposed to represent the last common ancestor of all humans, maternal ancestor of all mitochondrial DNA molecules on the planet, and Y chromosome Adam, which is basically this individual we can trace, all males can trace their Y chromosome back to. Okay, and again, the data here, both on the mitochondrial data and the Y chromosome data, basically support this idea that all humans on the planet can trace their ancestry back to somebody who lived in Africa, and it supported the idea that humans, all modern Homo sapiens, arose in Africa, and that much more recently, some of them left Africa and migrated out to other parts of the planet. If you should take a 23andMe test, it not only gives you information about your nuclear genome, but it also gives you information about your mitochondrial genome. So typically it organizes the mitochondrial genome into what they call a mitochondrial haplotype. And basically, mitochondrial haplotypes are just a set of shared polymorphisms that distinguish different populations in the world. The haplotypes typically have letter names associated with them, and the letters don't really convey anything other than that you know they were basically given the letter in order in which they were identified. So A is the first haplotype that was identified, B was the second, so on and so forth. But based on the haplotype, you can actually sort of say something about what the origin of your mitochondrial genome is. Well, all of our mitochondrial genomes can derive from a population in Africa, but the mitochondrial haplotypes that are, for example, if you look in Africa here on the map, the ones with the L, M, or N haplotypes, those are ones that are actually very similar to African, more commonly found in African populations. The ones with other letters are sort of distinguish different populations in different parts of the world. So basically, based on your haplotype, you can kind of get a sense of what the origin of your mitochondrial genome is. Again, we can all trace our ancestry back to Africa, but the haplotypes that are not that are the ones out of Africa are ones that are just different a little bit from the ones that were in Africa. And each of these haplotypes are just distinguished from one another based on polymorphisms that distinguish the different populations. Okay, so I'm going to talk a little bit about more of the evidence for the out of Africa model, because it turns out you can use mutations to track population migration as well. So I'm going to give you a very simple sort of simulation to kind of give you a sense of how this works. And I just want to there's a few assumptions in my simulation. The assumptions seem to fit, what we know about human evolution anyway, so it's not, I don't think the assumptions are any problem for what I'm about to tell you, but I just want to make sure I'm clear on what the assumptions are. Basically, one assumption is that only a small portion of the population migrates, because I'm going to show you my population migration. And the other assumption is the populations remain stable over time, or that they grow at roughly the same rate in different locations. All right, so I'm going to run a simulation now for you. Okay, so let's say this is your starting population, and these are different sequences of the same gene. Say, for example, in the starting population, could be a portion of the mitochondrial genome. It doesn't really matter. But let's say the starting population, everything has the same sequence. And then I'm just going to run off time in the bottom. I'm going to track time by these little dashes that I'm going to run along here. So let's run off a little bit of time. And as you know, as the genomes are being replicated, new mutations will arise in the population as a result of rare replication errors that occur at each generation. So we'll run off a little time. And then over this period of time, some new mutations have arisen in the populations. Now all the molecules are the same. We'll run off a little bit more time now. A couple of new mutations have arisen. We'll run off a little bit more time yet. Some additional mutations arose. And now I think I'm going to run off a big chunk of time here, and we'll have a bunch of new mutations that accumulate over time. And we'll just assume these are all neutral mutations. They're just mutations that are accumulating over time as the genome gets replicated at each generation and new mutations arise. Okay, so now we're going to do something different. Let's just say this is a population that's all present in one part of the one part of the planet, let's say. And then we're going to have a subportion of the population migrating out of that starting population to some new location. Let's say that the starting population is Africa, and we have migration out of Africa, say to the Middle East or something like that. Okay, so a couple of conclusions we can draw is that these mutations that are present in the starting population, they're going to be more common in the new population because only a subset of the diversity that's present in the starting population is going to leave, okay? So these mutations that are, or these polymorphisms, whatever we'd like to call them, that are present in the migrating population, they were also present in the starting population, but since only a subset of the starting population left, only a subset of the genetic diversity left with it as well, okay? And so the ones that actually leave the starting population, these are called founder effects. And it turns out founder effects are responsible for higher frequencies of certain diseases or traits in subpopulations. And you see this all over the planet. Here's a couple of known founder effects. So one founder effect is that there's a large clan of related individuals that have this disease, Huntington's disease. And this is really a terrible disorder. I think I mentioned this earlier in the course. Basically, it's a neurodegenerative disease. It's a dominantly acquired neurodegenerative disease. Typically strikes in your 40s. It impairs you cognitively and eventually leaves people dead, typically by the time they're 50 or something like that. So it's a really terrible disorder. And you don't really have any sense that you have this until you reach your 40s typically. So it turns out there was a large clan of related individuals that had this in a portion of Venezuela, the Lake Maracaibo region of Venezuela. I may not be pronouncing that properly. And I showed you this image on one of my earlier lectures. This is actually a picture of somebody, Nancy Wexler is the name of the person that's pictured here, where she's actually showing a pedigree of all the related individuals in this region. And the pedigree is so large because so many people have this disease in that region that the pedigree basically spans across the entire wall and goes around a corner. But basically, again, this is thought to arise because somebody actually migrated there that happened to have Huntington's disease, and the small population that arrived there had Huntington's disease, and that gets spread throughout the population very quickly because it was just present at a higher frequency in the starting population. So just a little bit of a side, Nancy Wexler is actually a very interesting person. Turns out she has Huntington's disease that segregates in her family. Her mother died of Huntington's disease. And she never disclosed for many, many years whether she was somebody who had Huntington's disease. And it was impossible to know that until the gene was identified, which she played a large role in identifying that gene. She was the main person who spearheaded the efforts to identify the causative gene. But she's recently revealed that she, in fact, does have Huntington's disease, although it seems to be a fairly mild form because I think she's in her 70s now, and she's just recently revealed this. And she gave an interview, I think it was published in the New York Times. Very interesting person, kind of a sad story that she has this, but I don't think she sees herself at all as a victim. I think she feels like she had a really productive life, and anyway, she's a very upbeat person. So it's an interesting story if you're interested in hearing more about this. Okay, but I diverge from the main point here. Just briefly, another founder effect that's the focus of a big study right now. There's an extended clan of about 5,000 people in Medellin, in Colombia, with an early onset heritable form of Alzheimer's disease, typically striking people in their 40s. And there's a number of clinical trials going on there using treatments for Alzheimer's disease, simply because you can identify these individuals with a simple genetic test long before they have symptoms. So people have tried doing clinical trials for Alzheimer's disease for many, many years. They've all failed. And the main reason they failed, people think, is because typically people don't get treated for Alzheimer's disease in these clinical trials until they have the disease itself. And so the thought is if we can treat them before they develop Alzheimer's disease, maybe the treatments will be more effective. So we'll see. And there's lots and lots of examples. I can go on and on here, but let's get back to the point. Okay, so these mutations are more common in the population that just migrated. And now we're going to run off a little bit more time here. So what you're going to see is some new mutations arose in the starting population, and there's new mutations that arose in the migrating population. And if you look at the new mutations in the migrating population, those are unique to the migrating population. And there's also going to be some new mutations in the starting population that are unique to them because these populations are not mixing with one another. Okay, so now I'm going to put another migration on here. So now we've got the newest population. All right. So I can ask you a few questions here. So which population has most variation? Which has the least? Hopefully you can see that actually it's the starting population that has the most variation. If you compare the sequences, you're going to see a lot more variation in the sequences in the starting population because they've just had a lot of time to acquire all that variation. The one that has the least variation is the population that's migrated the furthest because always a small subset of the variation that travels or migrates typically. Another thing you can sort of tell is that you can sort of tell that which populations are migrating because new mutations are occurring in the populations that are migrating that aren't present in the starting population. So you can kind of track movement by looking at the occurrence of these new mutations that are present in the migrating populations. Okay, so a few general conclusions that we can draw from my simple simulation. Stable starting populations will typically have more genetic variation than small migrating populations. Populations that migrate the furthest will have the least genetic diversity, and new mutations will arise at the same rate in starting and migrating populations. And it's actually that helps us to track which populations are actually migrating. Well, there's actually all features help track migrating populations. So again, all these features will allow us to ask when and where modern humans arose, where they went, and when they went there. But you can also apply these same principles to like population migration for pretty much any population that you're studying. It doesn't have to just be humans. Okay, so using some of the things I just talked about, let's go back to this idea that modern humans arose in Africa and left Africa. So it turns out, let's just talk about genetic diversity in particular. If we compared mitochondrial DNA sequences between non-Africans and between Africans, what you'll see is, just making pairwise comparisons, that there's more differences on average between African mitochondrial DNA sequences than there are between non-African mitochondrial DNA sequences. So in other words, what this is telling us is the reason there's more differences between mitochondrial DNA sequences between Africans is because they've had a lot more time to acquire all this genetic diversity, whereas if you make the same comparison between non-Africans, you don't see as many differences, simply because we haven't had as much time. People who have left Africa haven't had as much time to acquire all this genetic diversity. This again supports the out of Africa model of human evolution. It says that humans arose initially, modern humans arose in Africa, and more recently they migrated out of Africa. Okay, so there's also some nuclear DNA evidence in support of the idea that that all modern humans derive their ancestry to Africa. It turns out African populations have about 15% more nuclear genetic variation than non-African. Again, this is again consistent with the idea that there was more time to acquire all this diversity before a small subpopulation left Africa and migrated out to the rest of the world. Turns out more than half of this variation derives from the founding population. In other words, it was all present in Africa before anybody left Africa. And this is not really related to the out of Africa model, but I just wanted to mention that about 85 to 90% of the most common variation is shared across all populations in the world. Okay? The main point here is that there's a lot more variation in Africa than there is out of Africa. Okay, this just leads me to an interesting hypothetical question. So there's a science fiction film that was it's actually sort of before my time, to tell you the truth, but there's a science fiction film that was from the early 1950s, I believe, 1951 or something like that. I've seen the movie because I'm a science fiction fan, but I'll just tell you the plot. The star Belus will collide with Earth and wipe out all of humanity, and some philanthropists have decided to donate some money to build a rocket ship that will take a select few people to Zyra, a planet which may be habitable for humans. And they've had to choose a bunch of people that they think would be best to reflect the human race. So the question is, who would you choose to represent the human race? And I can show you an image from the movie. This is an image that shows the individuals that were selected for this. And I think if you look at this and you listen to what I just got through saying, you'd realize that if you're thinking of this from a genetic standpoint, this is not really probably the population of people you'd want to be present on this spaceship to reflect all the variation on the human race. As I already mentioned, most of the variation that exists in the human race is in Africa. So you'd want to have a lot of people from Africa on this spaceship too, or else you're going to lose most of the genetic variation that exists in the human race. Anyway, just sort of a interesting, way of how people thought about things back then. Okay, so how does all this stuff that I've been talking about fit with the fossil record? It turns out, if you look at the fossil record and you ask, what are the earliest known fossils of anatomically modern humans? Basically, they date to about 200,000 years ago in southern Ethiopia, roughly. So it turns out the fossil record supports an African origin of all modern humans that goes back about 200,000 years, and all the genetic evidence also supports that model as well. So basically, everybody on the planet can trace their origins back maybe about 200,000 years to a population that existed in basically southern Ethiopia and Africa about that time. So kind of interesting. So and the data that supports that is basically mitochondrial DNA, Y chromosome, and there's nuclear evidence that supports all of this this out of Africa theory. So I'll finish this lecture on a topic that's related to the very first slide and the very beginning of this lecture, and it's going to be regarding a recent example of using mutations to track species migration, and the specific example is COVID-19. So just a little bit of background information about COVID-19. So it has an RNA genome. This is not atypical of viruses. The genome size is about 30,000 base pairs, and it's thought that COVID-19 acquires about one new mutation every 2 weeks. So it turns out RNA viruses have much higher mutation rates than, say, DNA viruses. And that has to do with something I didn't really have a chance to talk about in this course, but it's kind of an interesting topic. But basically, DNA polymerases, they actually occasionally will insert the wrong base, but most of the time when they insert the wrong base, the polymerase pauses and it actually removes the base before it continues to synthesize a new strand of DNA. RNA polymerases don't have the ability to actually pause and remove the wrong base. So when they insert the wrong base, which they do at probably about the same frequency as DNA polymerases, they can't correct their mistake. They just continue to move on. So that's why they have a higher mutation rate than, say, DNA polymerases. Okay, so basically, people have been trying to understand the spread of COVID-19, and the way they've gone about doing this is that they basically collected COVID-19 samples in different locations in the world at different points in time. So, for example, there are some that have been collected in China, some in Europe, some in the US, and at various other locations throughout the world. And they were collected at different points in time. And then the viral genome was sequenced, and that's actually something you can do fairly easily now. So if this arose, say, 20 years ago, this would be a very difficult thing to do because the sequencing technology just wasn't as good back then. It was very expensive to sequence genomes, very time consuming. Now we can do it very cheaply and very quickly with next generation sequencing approaches. And then based on the sequences, and again, because the viral genome acquires mutations at a high rate, you can basically use mutations to construct an evolutionary tree. So they basically used algorithms for doing that, and they just grouped the genomes according to similarity to create their evolutionary tree. This is one of the trees that was created using this approach. This is work that was carried out by Trevor Bedford at the Fred Hutchinson Cancer Research Center right across town. And I don't want to imply that Trevor Bedford is the one who's done all this work because there were many people that were involved in collecting these samples. Many different labs were involved in sequencing the viral genome. But this is a genetic tree or evolutionary tree that was constructed based on the sequences of these viruses in different locations in the world by Trevor Bedford. And just a few points I want to make on this tree. It's a nice, pretty set of colors on here, but the colors all mean something. I've actually placed a link to Trevor Bedford's lab because you can actually sort of access this tree, and there's various parameters you can play around with to understand more about how the tree was constructed and to understand more of the details in the tree. But I'm only going to make a couple of points here at the very end of this lecture. One of the points is that this tree was just constructed with sequence information, and the interesting part of this is more of a reality check. All the earliest branches arise in China, so that's basically just supports the Chinese origin of COVID. COVID should say COVID-19. I've got a typo in my slide here. I'll correct that later. But the point is that this is really more of a reality check because I don't think there was any uncertainty about the origin of the virus. But it's just interesting that the evolutionary tree that's constructed based on this information also matches what we actually know. So that's more of a reality check, like I say. The later branches in the tree are mostly outside of China. So again, this is all color-coded. The purple color represents branches that were in China. The other colors represent other portions of the world. So all the later branches are not, you don't see any purple here because these are samples that were collected outside of China, and these are basically later branches in the tree. So they're evolutionary descendants of the ones that existed in China initially, because they have mutations that track to these different locations in the world. The red tips, these little red balls, are mostly sequences that were obtained from samples of the virus that were collected in the U.S. And the red balls are actually represent North America, so some of them are from Canada, some from Mexico, but most of them are from the U.S. And then if you look closely at the red balls, what you can kind of see is that they kind of cluster mainly in two different locations. There's this top location and this sort of lower location. And so it turns out the top location, those are US cases that probably came from Europe because if you look at the nodes of these trees, those are European nodes, the sort of the far left side of the tree. So they're later branches of this whole evolutionary tree where the virus moved from China to Europe, and then this implies that the virus that had moved from China to Europe came from Europe to the US, basically. And then this lower cluster of US cases, those are probably cases that came directly from China. So the take home from this analysis is that it turns out most of the US cases of COVID-19 probably came from Europe, not China. Now, of course, all the cases originally arose in China, but the point I'm making here is that of the cases that arose in China, only a small fraction of them actually ended up coming to the US directly from China. Most of them, particularly those on the east coast of the US, came from China to Europe and then from Europe to the US. Now, of course, this is an article in the New York Times that basically first reported this finding. This is actually, the Times isn't the one who reported this finding. It was actually reported in a scientific by a scientist, basically, and I think it's now published. But but this is an article in the New York Times that first reported this idea that most COVID-19 cases came from Europe. And so I've got a link to the article if you want to read the article. The article also cites a couple of different labs that do this conclusion. One of them is actually shown in the very beginning of the article here. But I just wanted to make a point that we sort of live in interesting times now where it turns out politics oftentimes trumps science these days. So it was just a couple days after this article was published in the New York Times where our president tweeted that the fake news New York Times is tracing coronavirus origins back to Europe, not China. So he seemed to express some bewilderment about this point. And then interesting, he also sort of said, I wonder where the failing New York Times got for this one. Are there any named sources? But the funny part about that is that if you look at the very beginning of the article, there is in fact a named source. They basically cite right at the very start of the article that it's a scientist at the Icahn School of Medicine at Mount Sinai who actually is responsible for this study. So again, I'm not bringing this up because I know that there's a lot of political divisions in the country, but the point here is Donald Trump. It is true the virus arose initially in China, but that's not the point of this article, and I'm sure he probably knew that as well. The point of the article was just to say that the cases that came to the U.S. mostly came through Europe, not directly from China. Anyway, so we live in interesting times, so just thought this is an interesting point. All right, so that's it for this lecture, and we have one lecture to go. The very last lecture probably won't be quite as long as this one. It's going to be mostly related to some interesting topics in human evolution. Okay, that's it.